Okay, so in agriculture, what are the examples that we have seen? Golden rice. Golden rice we saw. We also saw what did we say? BT and RNA interference. Okay, yeah. BT and RNA interference. So agriculture, these are the three major examples. So there is also two more which is mentioned in package. I'll just give you the name of the plant and what manipulation, how is it genetically modified. So that much is enough. So once you are done with this, we'll go to that. Next is medicine. Yes, ma'am. Then how double stranded RNA will form? Already one will be present in the nematode only, but we have to make sure that while it is entering the nematode, it is already going to immediately start producing the double strand. Okay, just to ensure that the double strands are already going to be there to immediately trigger it. Okay. So this is not only going to kill already existing nematodes, it is also going to prevent further new infections also. I'm getting very sleepy now. Completed? Sveda, fast this way. Get the point. Okay, so the next two more examples will be same is flavor saver tomato. Okay, so next what we are going to see is flavor saver tomato. So this flavor saver tomato here, gene was not added. This is an example for gene deletion. The gene that is deleted here is for production of polygalacturonase. So polygalacturonase production was stopped. Why? Because this is an enzyme that is going to cause faster ripening of tomato. So why is this bad? Post-harvest losses. So tomato is going to be harvested, stored in one place and then transported to different places. Might take 3 to 4 days. What is the shelf life of tomato? 2 days. Okay. So it will go back. So now what was done is removal of polygalacturonase. Okay. So the gene that is producing polygalacturonase enzyme, that is removed, which means it can stay for 5 to 7 days. Because already 4 to 5 days it has been transported. Okay. So that is because of this removal of this gene only, which is for polygalacturonase, which causes ripening of tomato. Why flavor saver? Is because it's found that after removal of this enzyme producing gene, the flavor of tomato also increases. So it's called flavor saver tomato. Okay, so this is just one more example. So far we have seen how many examples for agriculture: golden rice, BT plants, RNA interference, and this tomato. Okay, so next is use of biotechnology in medicine. So first thing we'll be seeing about is therapeutic drugs. what is a drug? What's a drug? Can anyone tell me what is a drug? Medicine. Okay. So why drugs are called as drugs then if they are medicines? Okay. So anything that causes physiological, psychological changes in your body will be called as a drug. So therapeutic drugs. So these drugs are produced through biotechnology because the benefits are mass production. As we already saw in the previous lesson through bioreactors. So mass production, that is production in a large scale. Means larger population can get it and it can avoid unwanted immune reactions. What is it referring to? If you want some enzyme or protein, you can extract it from other animals and introduce it into your body. But since this is from other animals, there may be other proteins also present which your body will identify as antigens and it is going to produce immune reactions against it. Very severe reactions may also be possible. So we can avoid it if it is going to be produced through biotechnological methods. In that case, the first thing we are seeing is therapeutic drugs. Around the world, how many are there? Around the world, there are 30 therapeutic drugs present. But in India, how many are being used? 12 therapeutic drugs are used. We'll see the name of a few of them. So first is going to be human insulin. Against what is human insulin used? Obviously, yes, diabetes mellitus, and then human growth hormone. And another most commonly used one these days is FSH. For what FSH is used? Fertility treatments, okay? In fertility treatments, FSH is going to be used. And another most common one that is being used in India these days is streptokinase. What do you know about streptokinase? Yes, clotbuster. So what is a clotbuster? So in your blood vessels, if there are any clots that are formed, it can dissolve those clots. Okay, so clot buster. So other heparin is produced that way. Erythropoietin is produced through biotechnology. What is erythropoietin used for? And to increase RBC levels, treat anemia. Okay, so erythropoietin. And there is also granulocyte colony stimulating factor. This is very important. This is for cancer treatment. Do you remember immunotherapy? Where we are using biological response modifiers. Okay, these all come under that. So granulocyte colony stimulating factor, which is called as, that's for cancer treatment. So now these are some of the common ones that are used. So now we are going to see among these production of one in detail. Okay, that is what? Human insulin. So human, how it is produced? What is it used for treating? It is used for treating adult onset diabetes mellitus. So this is for insulin dependent diabetes mellitus, mostly coming under type 1 because this is going to be a type of autoimmune condition, okay, where the own cells are going to attack your B cells, so there is no other way the person has to take insulin. So now, before biotechnology, what was the source of insulin? Porcine pancreas. What does porcine mean? Pork, pig. Bovine means what? Bovine, cow. Okay, so bovine means cow, porcine means pig. Okay, so porcine pancreas was the source of insulin. But it is very difficult. Why? Because around 4,000 kilograms of pancreas has to be used to extract just 250 ml of insulin. And that too not in a purified form, because introducing it in people triggered severe allergic reactions, immune reactions also. But now we are producing it in mass production, large scale production in tons, because now we are producing it using what? Which organism? E. coli, yes. So which is going to be extremely large scale. So that is what we are going to see. Now, which company did it? That is again quite important. So who was the company? Eli Lilly. In which year? That year is important. Anybody knows which year? 90? 1983. 1990 something happened. Anybody knows in this lesson only? Gene therapy, adenosine deaminase. Yes, once the lesson is over, we'll revise all the years of both the lessons. Important. Okay. So 1983, a company called as Eli Lilly, they found a way for producing humulin through E. coli. But what is the challenge here? Why is this very big? So now we will see a little bit about human insulin. So normally how we will do it? How a biotechnological product will be formed? You will take gene from the source, you will introduce it into a plasmid, put it in E. coli will start secreting it. We can't do it like that here, because the human gene for insulin is going to code for what? Proinsulin. So it produces only proinsulin. What is proinsulin? Similar to protoxin. So this is inactive form. So we should understand the inactive form. Inactive form, what it is going to have is, it is going to have an A chain, it is also going to have a B chain, but in between A and B chain, there is a C chain. So inactive proinsulin, it is just going to be one long polypeptide chain. Please listen, one long polypeptide chain. Even though there are two different chains A and B present, they are only separated if you remove C. Okay, so there is going to be an A chain, there is the B chain, and then the C chain. As long as C is connecting these two, isn't this just one whole protein that looks like this? Yes, so unless you break C, so now which means if you introduce this gene directly into E. coli, what E. coli is going to produce? Proinsulin. So then what is the problem? We can remove C, right? That is the difficult part. Removal of C outside a living system that has the procedure to know, that has the enzymes to break it, is not there in E. coli. Okay, so only human cells can produce the proteins and enzymes that can break these peptide bonds, which E. coli doesn't have. So it is very difficult. So what did Eli Lilly do? We will see it first right under this. Understood why is it difficult? Like directly you can't put it inside the gene, you can't simply take and put it inside E. coli because E. coli then will start producing only proinsulin, which is of no use in our body. Proinsulin and? And normally then how is insulin given? What is the difference going in vein, going in muscle? Everything anyway mixes with blood only. How will that change?
Hepatitis vaccine, how will they do that?
Now, so now what happens then? What did Eli do? That is very important. So before that, we'll see the bonds that are present also. So what bonds are present between A and B? Disulfide bonds are present. So I'm just writing disulfide bonds like this. And here also there is a disulfide bond within the HA. So this is what? Proinsulin. Okay. So now Eli Lilly, what they did was, this is a limitation, one long polypeptide chain, which means what? It directly, the gene for proinsulin directly cannot be inserted into, please write now this since we already discussed it, and it cannot be directly inserted into E. coli since E. coli lacks the enzymes needed for removal of C chain. Okay, so that is what they did. So now here, this is what we call as the proinsulin. So now if the C chain is removed, what it will look like? The A chain is going to be there, B chain is going to be there, and is there any change in the number of disulfide bonds? That there is going to be no changes. It is just going to be the same. So it is the A and B chain. So remember that between proinsulin and the humulin, which is the human insulin, there is no change in the number of disulfide bonds. Will there be change in number of peptide bonds between pro and normal insulin? Yes. C chain is gone. Okay. So it means there will be difference in that, but this is going to be not changing human insulin. Okay. So now what Eli Lilly did? How do you think they produced this insulin in an E. coli? So what they did was, the DNA sequence, so this was what was done by Eli Lilly. The DNA sequence Please listen. The DNA sequence for A and B chains, they were individually chemically synthesized. Of course, the template, the sequence was identified from human gene only, but they didn't just take that whole gene. They saw that sequence for A chain. They chemically synthesized that sequence, and it's easy. You just have to add the bases in that same order. That is all. So they chemically synthesized the gene or DNA for A and B chains individually, separately. Okay. So which means what? Let us see what is happening in A chain here. Let us see what is happening in B chain here. So A chain's own gene was made, chemically synthesized. So this is going to be the DNA which is going to code for A chain. Okay, and then which means for B it is going to be another gene. Okay, so this is going to be the DNA that codes for B chain. And now what happens? We are going to introduce it into E. coli. How did we introduce it into E. coli? Through a vector. Through a vector, we introduce this gene into E. coli separately, and this was done in another E. coli separately. Now what did they produce? Once the plasmids started multiplying, these all started producing a lot of A chain, which had 21 amino acids. And then here this E. coli produced B chain, which has 30 amino acids. So in total, mature human insulin, how many amino acids are only there? 51 amino acids are only there. Okay. So now then what happened? Now we are just going to allow them to form disulfide bonds. Okay. So when they are mixed, they are going to be allowed to form disulfide bonds. Sulfide PHIF. Disulfide bonds, which gives us what? The mature human insulin. No extra steps necessary, no extra enzymes need to be provided. It is not going to have any effects also, no immune effects. So this is how Eli Lilly produced it. Okay. So synthesis of insulin in a very large scale occurred in this. Okay. Understood? Please write. From here, next we will see gene therapy. Done? Okay. Shweta?
Fast.
Okay. So next is gene therapy. Therapy indicates what? Therapy indicates treatment. Okay. So here we are going to see for the genetic disorder ADA deficiency. Now what is ADA? ADA stands for adenosine deaminase, which is going to be an enzyme. So this adenosine deaminase function we'll see, you can write it as simple. So please write, so adenosine deaminase is going to be an enzyme that breaks down what? From the name, what do you understand? Sudesh, are you there? Adenosine deaminase, it's an enzyme. What do you think it breaks? Yes, so it is going to break adenosine, enzyme that breaks down adenosine, because adenosine is a toxin that will destroy lymphocytes. Again, complex mechanism, very much simplified. So it is going to destroy lymphocytes. Okay, all lymphocytes, every single lymphocyte in your body will be destroyed if a person is having adenosine deaminase. Now, where did this adenosine come from? In your body, cells are breaking down. Constantly cells are destroyed. Your own cells, when they are getting older, they are destroyed. What happens to the nucleus? Also destroyed. What happens to the DNA? Broken down. It turns to nucleotide. Nucleotides are broken down, nucleosides. They should also be further broken down to nitrogen bases. But for breaking adenosine, this enzyme is needed. When that enzyme is not there. Now, why is it not there? Why is there a deficiency? It's a genetic disorder. Means what? The gene for producing that enzyme is missing. Okay. So in that case, it destroys the lymphocytes. It's a genetic disorder, which means gene is not there right from birth. Which means this is a disorder that occurs right from birth. So it's a disorder. What type of disorder is genetic? Please write. It is a genetic disorder where what happens? There is going to be no gene for ADA. Which means right from birth, what is not present in the child? There is no immunity. Now this type of disease, ADA deficiency, leads to something that is called as severe combined immunodeficiency. Which is referred to as SCID, also commonly called as the bubble boy syndrome. Why? Because the child born with this is always supposed to be staying inside a sterile environment. So the air that is given, the food that is given, everything should be 100% sterile. Okay. So now, if this child is exposed to a pathogen, what will happen? No matter how simple that pathogen is, the child will exceed. We call a pathogen as simple. Why? Because your body can easily form immunity against it. That's not simple. So if your immune system is absent, how is it going to protect you? It will not. Okay. So that is what is happening here. Now, why is it called a bubble boy syndrome? Why is it a child always? Because they don't give to turn into adults, because no matter how much you are going to protect their environment, some way, somehow pathogen can enter inside. So now, which means sure mortality is there, but there is a treatment. There are treatments available. So now, what are all the different types of treatments? So this is treatment only. Cure is in only one way. We'll see what that is. The first treatment is going to be enzyme replacement therapy. So enzyme replacement therapy, it is like insulin only. So your body is not producing insulin, so you have to inject insulin. Same way here, the body is not producing an enzyme, so it is going to be injection of enzymes. Injection of which enzyme? ADA. But this has to be done constantly, regularly. So it's not a long-term solution. Second solution is what? Bone marrow transplant through gene therapy. This is what gene therapy is. So the gene therapy was first done when? It was first done hope you are all writing. So it was first done to a 4-year-old girl in 1990. When was Eli Lilly insulin? 1983. 1963 what happened? Two enzymes. Okay, so this is 1983, and in 1990 the first gene therapy was done to a 4-year-old girl. Bone marrow transplant. Now what is a bone marrow transplant? Enna pannuvaanga? What do you understand? Soumya, are you there? What's bone marrow transplant, Soumya? Okay, that is what we normally assume, because kidney transplant na where or kidney eduthu we are putting, but that is not what bone marrow. You can't scrape eppadi eduthinga bone marrow veliye. It's not that. You take cells from bone. And it is not from donor. It is from the. It is from the girl's own bone marrow. Okay, there is no donor here. That is for different condition. Okay, so in this case it's not there. So how is this done? So this is a very simple step. So first, stem cells, which is basically just lymphocytes only, means immature lymphocytes from the patient who is suffering from this disease. So where are these collected? From their bone marrow. Okay, first step was simple. We are collecting whoever, let us say this 4-year-old girl's bone marrow cells. And then what we are doing is, we are introducing, this step is very much important. We are introducing the cDNA of the ADA gene into the lymphocytes. So many things are happening in this statement. So what are we doing? We are introducing cDNA. What is cDNA? Who can tell me what cDNA? Complementary DNA. Okay. What do we mean by that? Good, yes. Okay. So introducing cDNA of ADNA into the lymphocytes. Now, collected from the bone marrow, and these lymphocytes, these are allowed to multiply. These are cultured in the lab, we'll say. Means more copies of them. Okay, so cultured in laboratory conditions. That is lymphocytes after extraction. Now, cDNA. cDNA is, please listen now, don't write. It is the complementary DNA. What is a complementary DNA? This is what is produced when, again, please listen first. This is what is produced when the gene, the actual gene is taken. That is in the form of what? DNA only, right? So it is allowed to form an mRNA. And then this mRNA what we are going to do, what we are going to do is, we are going to turn this mRNA back into a DNA that is called as cDNA. Why do this? What is the purpose of this? அது அங்கேயே இருந்திருக்கலாம், அதுக்கு நம்ம உடம்புல இருந்திருக்கலாமே. Why we are changing this? What is the point of this? Okay, when you say a gene is there, coding for a character, every single part of the gene is coding. It will have both introns as well as exons. Just the coding part, exons alone. Introns தேவையா? தேவையில்லை. And introns, if you include introns also, the gene becomes very big. Now what we are doing is, we don't want that introns anyway. So we are turning it into mRNA. But now what sequence alone is going? Exons are also going. When you turn it back into a DNA, the whole gene is going to be now compacted with only what? The exons. It is going to be easy to regulate, easy to control, easy to introduce into these cells. Okay, so that is why we are using a cDNA, a complementary DNA. Essentially, that means only what is present? Only exons, only the coding part. Easy to control, easy to regulate, easy to put into a cell, and faster it is going to be showing immediate responses. Okay, so that is why we are using ADA cDNA. So that is the reason why ADA cDNA is going to be introduced into the lymphocytes. Please write now. Now after that, what the lymphocytes start producing? Since now the gene is present, the lymphocytes start producing ADA, and this is what is introduced into the patient. So now what we now have, there are now a lot of cells circulating in the girl's body which is secreting ADA, which means immune system functions normally. So now what happens is, since ADA is now going to be produced in the patient's body, but is this a permanent solution? Why? This is not permanent because lymphocytes are mortal cells. Means what? They will die after their lifespan is over, which means again it has to be done. Which means it is a long-term solution compared to enzyme replacement therapy, but not a permanent solution. But what would be a permanent solution? Doing this in embryonic stage. So, இது வந்துட்டு second is bone marrow transplant over, and the third step is what? The third way is gene therapy, that is bone marrow transplant done in embryonic stage. Anga we can't call it as bone marrow, and idum bone marrow form agala. So done in embryonic stage. Okay, so this is the what? This is going to be the cure for ADA deficiency or any type of genetic disorder. Okay, so this is going to be gene therapy. So next part we are going to see in medicine is diagnosis. So this is our gene therapy. Next is Mushtiama gene therapy. So next what we see is molecular diagnosis. What is that?
Identifying diseases.
Identifying, diagnosing diseases in very early stages. Why would we need that?
To prevent war.
To do what?
To take precautions against the disease.
To take precautions against the disease. Okay. RM3 students, can you all raise your hands? RM3 alone, please. All RM3. Hi, all listening. Half of you are not raising your hands. RM3, okay. 8 only have raised. 11, okay, 11. Okay, ma, put down your hands. Thanks. Thank you. Okay, I'm now coming to molecular diagnosis. Who would want to do early diagnosis? Okay. Molecular diagnosis is for early diagnosis, which can give a much more effective treatment. So we can reduce the effect of the disease in our body. So now let us say, randomly will you go and test for malaria, typhoid? When you will test for typhoid? When symptoms are there. Okay? When only symptoms are there. So now what do we mean by early diagnosis is diagnosis before symptoms. So diagnosis before symptom and also to understand the pathophysiology. What is that? How the disease would affect the body. Okay, to understand the pathophysiology. Again, I'm telling you, if you don't have symptoms, you won't go and test for a disease, right? Because every day you are exposed to so many different pathogens. Why would you go and test for all of those? Under what context you will go and ask them to check for TB or check for malaria? We won't. Okay. So then, what type of diseases are we talking about here? Cancer. So, if there is a history of cancer in the family, or you feel like you have been exposed to some carcinogen for long unknowingly, in that case, you will go and test for cancer to identify the mutated gene, to check if it is there or not, even before the cancer shows up. Okay, because it will take a lot of time for the gene to actually get activated and form cancer. So, in that cases, but mostly if there is a case history of cancer in the family, in that case, a person can do early diagnosis. Okay, what else? HIV, yes. So if a person has done something, now they are scared that they may have HIV. In that case, they can't wait until the symptoms show up because we know that it can take up to 10 years also. So, but in a short span, if they start taking medication, they can prevent these symptoms. They can live a normal life, almost normal life. So in that case, it can be detected. So we are going to see what are all the ways in which, and even for COVID, when COVID was there, if someone has gone in through public transport or they have come in contact, even before the symptoms show up, because it takes at least 7 to 15 days for the symptoms to show up. So we can detect if the person has that virus or not. How? So first method is the most simplest method. Just by amplifying the DNA of the pathogen, we can find whether it's there or not. So for that, what we are going to use? PCR. So early diagnosis, first PCR. This is for amplification of DNA. Especially in early stages of infection. But PCR alone is not enough. PCR is just going to amplify the DNA. You should follow it up with other tests. Okay, it's just going to amplify the DNA. So, what are all the uses of PCR then? Detection, early detection of disease. Okay, so let's take that one. What else? We discussed some when we talked about it in the previous lesson. DNA fingerprinting, especially in cases of what? Forensic criminology, forensic studies. What is forensic? So in a crime scene, very less amount of DNA is left behind. It could be a blood or some tissue. Okay, so something that has cells in it. Other direct put you can't test. You need more copies of it. What if you lose it? Because it's very important information. So what is done? PCR is done. So forensic studies to amplify DNA on crime scenes. What else? Paternity. What is that? What is paternity testing? என்ன கண்டுபிடிப்பாங்க paternity specifically means what? என்ன parent? Mother யாருன்னு தெரியும் obviously. Mother வந்து தான் வருது. To identify who is the father. அதுதான் paternity testing. Okay, so to identify the father. Okay, so all these things PCR is going to be useful for. So yeah, so that is PCR. Can it be used in HIV? Why graft rejection? Why would we use PCR for that? If it is from another human, you obviously know it's not your own DNA, right? It means it will reject. Okay, HIV, especially in early stages, that we've already written, but anyway, right, specific for HIV, but it can only amplify, it can't identify whether it's there or not. Okay, so that is PCR. Second thing we'll be seeing about is ELISA. ELISA, we saw 3 different types of ELISA. What was it? Sandwich, what do you all remember? Direct ELISA, indirect ELISA, sandwich ELISA. We already saw how it works. So ELISA is, you can usually follow PCR with ELISA in case of HIV. Okay. So what is ELISA? It is an antigen-antibody interaction. So antigen-antibody interaction is going to be this ELISA thing. Okay, so here again early detection is possible because in very small concentrations also it is going to identify. And the third one is autoradiography. You would have studied, I guess, about autoradiography. What is autoradiography? Good. Yes. In whatever we are trying to find. Yes, good. So, autoradiography is usually used to detect mutated gene. You have to find out whether a gene is mutated or not. How we will do that? So first we will take cells from the organism or tissue where we want to check gene has mutated or not. And then we will do the DNA extraction. How we will do DNA extraction? However, we saw in the previous lesson. Okay, so we use the enzyme spooling. We have taken the DNA. And then we will fragment the DNA using what? Transcription enzymes. And then we will denature the DNA. That will only happen in PCR, right? We will denature the DNA so the templates are separated. Adukapura what we are going to do is, we are going to run it through agarose gel electrophoresis. While doing that, we are going to add a radioactive probe. Now this radioactive probe we are making. So a gene is mutated, right? We have to find out whether this specific sequence is mutated or not. That mutate actually we don't know. Okay. So what we will do is the probe that we are giving will be a single stranded DNA which will be complementary to the strand that we are trying to find. We know the original sequence. We know. But we don't know what the modified sequence is. So original sequence to complementary strand we are going to make, we attach a radioactive element to it. And then we run the gel. Now, if it is complementary, it will go and bind with it. Okay. So if it is not complementary, it won't bind, it would have flammable. So either one, we are going to blot it onto a photographic film. Photographic film and X-ray film will. So, which means all strands are now copied. So, radioactive element irundhuchuna, it will appear in that photographic film. Appadina enna artham? There is no mutation. But if it is not appearing in the photographic film, it means the complementary strand you have given has not bound. Which means what? Mutation has occurred. So, this is how we are going to use autoradiography used to detect mutated gene. So, first thing is what? DNA isolation. So DNA is taken from whichever animal, organism or human you want to check the mutation for. DNA isolation is done. DNA digestion is done. Digestion always means what? Breaking of bonds. Digestion. Okay. So DNA digestion is done and DNA denaturation is also going to be done. And then what is the next step? Okay, a complementary strand to the normal gene sequence. To the normal gene sequence, and this is along with an yes, so radioactive phosphorus or sulfur, so or sometimes iron, so radioactive component is going to be attached. Now this is called as what? This whole thing is going to be called as the radioactive probe. See, if it's normally a probe also, probe is usually a single stranded DNA complementary to whichever gene we want to identify. That is all. Okay. So, radioactive probe. When this radioactive probe is allowed to hybridize, with the sample DNA. What is the sample DNA? Sample DNA is the one that we want to identify whether it has mutated or not. So it is allowed to hybridize with the sample DNA. Now, if it is complementary, means the probe will attach. And it will appear what? Appear where? Appear on the photographic film, the X-ray film. Okay. So photographic film, which indicates what? No mutation. But
எப்போ முடிப்பீங்க?
நீங்க சாப்பிடுங்க. நீங்க சாப்பிடுங்க. என்ன சொல்லிட்டு இருந்தேன்? If it does not appear on photographic film, then what would that mean? Mutation has occurred. So if it does not appear on photographic film, it means that there is going to be mutation, which means what? Which means probe hasn't bound. So this is autoradiography. Other way also you can do it. Okay, so multiple ways this can be done, but this is the common way. Understood? What is this? Is it?
That's not.
medicinal use is done. So next we are going to see about transgenic animals. Break விடலையோ பரவாயில்ல முடிச்சிடலாம். Sudesh, what happened? Sudesh, your hand is raised up. அப்ப raise பண்ணதா சரி, okay. Done. Complete. Half an hour. Okay. Over. Okay. So now transgenic animals. Who are transgenic animals? What are transgenic animals? Why are transgenic animals? Genetically modified animals. What are all the common animals that are genetically modified? Spider. Cow. Liger genetically modified. Now tell cow. Did you say cow? Jessie. Rosie. Yamma, inga padam paathuட்டு irundhiya? Jessie. Okay. So cow. What else? Sheep. Mouse. Yes. Pig. Rabbit. Rat. Fish. Fish. Monkey கொடுத்திருக்காங்களா NCERT? Only these animals: cow, sheep, mouse, pig, rabbit, rat, fish. Okay, so anyway, majority of this is going to be mice. How much percentage? 95% of transgenic animals are mice. Why we prefer mice? Okay. What? That is what. Why here? Why not any other animal? Smaller, okay, and then which means what? Easy to regulate, easy to control, easy to grow, and also their reproductive phase is very less, which means we can observe the effects of gene manipulation in next generations also. In multiple generations we can observe in a short span of time. Okay, so since we can manipulate the gene easily, the effect also is going to be immediate. So that is the reason why mouse is going to be preferred a lot. So that is going to be transgenic animals. Now what are all the uses? Where, in what conditions we are going to use the transgenic animals for? Anyone can say? Medicine is tested. What type of medicines? Anything like vaccines or any other chemicals can be tested on them, and also we can induce a disease in them, and we can see how changes are occurring, and we can use chemicals on them to test. We can introduce genes that will make them produce some products that we want. Or if there is a new gene and you want to understand what its function is, you can introduce it in them and then see what type of changes their body is producing. Okay, so these are all going to be the condition. So first, what are all the uses as we see? The first use is study of normal development and physiology of that specific gene. Okay, so of a gene. So this gene can be introduced into the animal, and we can see what type of effect is going to be having on the organism, and just to understand the normal functioning of it and the effects that it is going to give on these organisms. Okay, so how these genes are going to be working is what we are going to find out. So here the most common thing, the example that is given for us is Introduction of genes for insulin-like growth factors. So we are going to observe what effects it is going to have on these organisms. Alright. So next is disease studies. Means what? We are going to introduce the genes for diseases and we are going to study it in these organisms. Now such are called as disease models. So such disease models are available for what type of conditions? Alzheimer's, cystic fibrosis, rheumatoid arthritis. One more is given. What is it? Cancer, I guess. We have disease models available for these diseases. Now, what exactly do we mean by that? Disease models? We have introduced these diseases in mouse and we are studying how they are responding to certain medications. What is Alzheimer's? What kind of disease is it? Memory loss. Okay, yes. Okay, so memory loss because of acetylcholine deficiency. What is cystic fibrosis? Severe and chronic memory loss means it occurs over time. What is cystic fibrosis? This is going to be extremely thick mucus. Usually it would occur in pancreas, lungs, sometimes in the intestine. And it will turn the tissues hard and fibrous eventually. That is called cystic fibrosis. Rheumatoid arthritis, I think you all know. What is rheumatoid arthritis? Joint inflammation caused due to what? It's an autoimmune disease where what happens? The immune system attacks the synovial joints. So now we have disease models. Means what? We have induced all these diseases in the organism. Okay. So what is? For production of biological products. This is where who comes up? Jessia, Rosie. So whatever products humans need, so those will be produced with the help of these organisms. So what kind of products are we talking about? First is a transgenic cow named Rosie. Formed in the year 1997. Okay. So what was Rosie capable of producing? She produced, very good, yes, human protein enriched milk. Okay. So produced human protein enriched milk, which is more suitable for human babies. And what is that protein called? That is alpha lactalbumin. How much was produced also you should know. So alpha lactalbumin, which was 2.4 grams per liter. Okay, so that was Rosie. And there was one more transgenic sheep. And this transgenic sheep produced alpha 1 antitrypsin. Okay, so this alpha 1 antitrypsin is used for treatment of what? Treatment of emphysema. What is emphysema? It's a respiratory disease, occurs only in people who smoke a lot. It affects the alveolar walls, it reduces the surface area for exchange of gases. Diffusion it is going to reduce. So biological products, and the next one is chemical safety testing. And the last one is vaccine safety. Completed under transgenic sheep. Now, chemical safety testing and vaccine safety is kind of similar. In chemical safety testing, what we are going to do is, we are going to make the animals more sensitive to a specific toxin. Make animals more sensitive to that chemical, and we are going to test the toxin safety on them. Means, what are all the side effects of using that chemical? Is it going to affect them a lot or not? So this is why transgenic animals are used. So that is how we make them more sensitive to that chemical. Vaccine safety testing is the same thing. So it is to test the safety of that batch of vaccines, which is going to be first tested on transgenic mice. Again, same thing, made more sensitive. So transgenic mice, and then it is used on monkeys, and then it is going to be tested on humans. Okay, so these are all the things that I use. For biological products, please add one more point for biological products. PKU, what is PKU? Phenylketonuria. So they are also trying for products to treat phenylketonuria, are also being tried by this biological products. Okay, so that is all. So these are all transgenic animals. Now the last part of the lesson. Yes. What, ma? In another 10 minutes, ma, not even 10 minutes. That's all over almost. We just have to discuss this bio patent, and then that is over. That is all. 5 more minutes maximum. Okay, ma. 5 more minutes. Okay, so next what we are going to see is the bioethics. Is it okay to do all these to animals? Who are we to do all these to animals? But we still do it. So to control the level of these manipulation both in plants and animals, because it is not only about ethics, so it is going to be forced upon animals and it is also going to mainly affect the ecosystem. We can't just introduce random genes and allow them to go in the wild. It will affect the ecosystem a lot. Plants also. So to control this in India, what do we have is to control RM2 students. There is no chemistry class for you today, ma'am. Ma'am is having some other class. So for you, there is no class today, chemistry. Okay. 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 So control. Okay, in India, what we have is GEAC, that is Genetic Engineering Approval Committee. What they are going to do? They are not going to allow you introducing any gene, any random gene into any animal. Okay, so there is a limit to what can be done. Okay, so that is purely controlled by the Genetic Engineering Approval Committee. So next is biopatenting. What is a patent? In general, what is patenting? It's like a copyright. Okay, so if it is your product, you are going to register it in your name so that others can't use it without your explicit permission. Now, initially, patenting meant for designs and models in engineering, things that you built. Okay, so that was patenting. So nowadays, you can do it even for your intellectual properties, like any songs that you make, anything that you produce on your own, anything that you write, anything. So, but what is a biopatent? This is specifically for organisms and the products they produce, and this is on a country level. So, if this organism and that product that it is producing, you have been using it for many years for a specific purpose, you can patent it. Okay, but this was misused by many countries. So, we are going to see about 3 examples for that. Starting first one is in the year 1997. Patenting of what? Patenting of a variety of basmati rice. Please write. So basmati rice. What variety? A company called Rice Stick. They said they have produced a new novel wild variety, a dwarf variety of basmati, by crossing it with a wild variety. They have said that they have produced a completely new variety of basmati, which means nobody else can grow this basmati rice anywhere. Okay, this or that are the new varieties they have produced. They can't produce anything. But the thing is that in India, how many varieties of rice is there? That is in botany. In zoology, it is 200,000. India has 200,000 varieties of rice, in which 27 variants are just basmati. So if they are going to patent that, what our farmers can't do, they can't grow those, harvest those specific type of basmati. So that happened in 1997. This patenting was eventually revoked because there was a lot of fight between both countries regarding this. And then 2 more things that they did was turmeric. It's what now? So what they did was, yes, so that is the important one. We have been using it for thousands of years. So the antiseptic properties of turmeric. This is now they sell this turmeric milk as golden milk. As if they only invented. Okay, anyway. So antiseptic properties of turmeric was patented. It meant that they only found, I think it was an European country. So They only found out that turmeric has antiseptic properties and they patented it, which means what? You can't put turmeric on your wounds anymore, they will arrest you, okay? They can put case on you. So that is basically what it meant. But then again, we have been using the turmeric for the same antiseptic properties for thousands of years. We have records of it, okay? So that was done. And then one more thing, all happened consecutively, I think in 95, 96, 97. So next was neem oil. No, it was contested and it was cancelled. Of course, it's not active. Neem oil, and how many billion people are there in India? What they will do case on all of this? Okay, so neem oil, its antifungal properties. Means if you get dandruff and if you put neem oil, you are not allowed to do that because apparently they only discovered it. So this is all biopatenting. Now this shows how one developed country can use this underdeveloped or developing countries. One more topic related to this is biopiracy. Now this is usage of organism or the products from these organisms and the traditional knowledge which will all be present in where only? Developing or underdeveloped countries only, okay? So in countries with what? Vast biodiversity, but they are financially poor, which means they are either underdeveloped or developing. And this is used by who? By a well-developed country, but it is poor in what? Biodiversity. Now they use this knowledge, okay, so they are going to use the traditional knowledge and they are going to monetize it, they are going to gain billions in profit. But what is not given? No compensation is given to that native country. Okay, so no compensation is given to native country. They don't even know that they are doing it. Okay, so no compensation is done. Or what is also not happening? First of all, there was no proper permission to use that organism because it didn't belong to their country, right? So no proper permission to use the products also. So that is called biopiracy. One example I'll just tell you, you don't have to write it. One example for this is in Africa, I forgot which country, there is a plant called as Pentadiplanta brasiliana. So it produces berries. In these berries, there is a protein called as brazil. And this protein, this is a protein, not a carbohydrate. I think it's somewhere around 2000 times more sweeter than the sucrose that we use. So what happened in American company, they isolated the gene for this brazil and then they introduced it into their corn kernels. So in their corn is the staple there in USA. So they introduced it in their corn kernels, they extracted this sweet chemical, they made corn syrup and they sold it as what? Sweetener for diabetic patients because it is a protein, which means what? It won't increase the glycemic index, glucose won't increase, but still the sweet taste is going to be there, billions of dollars. But to the native people who have been using the same berry for sweetening their food, did they get anything? No. So if they don't know that knowledge, this company wouldn't have gotten to know it. So that is what is biopiracy, okay, where country is using another country's resources without properly paying them any compensation is biopiracy. So with this lesson is over. With this, your portions are also finally over. Okay, so that's it. Please go for your lunch break. Thank you.